Okay, so we're going to be starting Unit 2. Unit 2 is biology. So we'll be talking about the units, the basic unit of life, the cell. Okay, in the same way that we started chemistry by looking at the basic unit of matter, the atom. Okay, so we start out small and then we're going to expand out from there. So we're going to start out with the cell theory. Okay, that is, how did we discover cells? Okay, who would have been important in that? Why cells are important? Why their discovery was important? Kind of our changes to our understanding of how cells work and what they are. Okay, that kind of stuff is what we'll be looking at, and then getting into looking at a microscope, because next week, okay, we will be doing the lab using the microscopes. I still have to figure out exactly how we're going to do that COVID safely. Probably going to involve like cleaning the microscope often. Okay? But yeah, we're still going to do it because we don't want to miss out on that part of it. Okay? All right, so what we're going to go over is cell theory and microscopy. So we need to know the points of the cell theory and how they relate to living organisms. Okay? There are 3 points to the cell theory, which is one less than the atomic theory, so you got less to remember. Okay. Know who contributed to the development of the cell theory, and recognize the implication of the similarities between plant and animal cells in terms of their evolutionary relationships. Okay. All right, so we got a picture of a basic plant cell here on the front page, okay, or on this first page. What are we noticing about this plant cell?
It has a cell wall.
Okay, it's got a cell wall. What else? If I was to put an animal cell beside it, would they be more similar to each other or more different from each other? In terms of like, if I had them together, would I see more obviously how much they have in common or how much was different? Yeah, more in common. Okay, plant cells and animal cells are so much alike actually, okay, that it's the reason we believe that everything on Earth came from a common ancestor. And if you look at the cellular structure of all living things on Earth, the basic structure of the cell is basically the same. Okay? There are going to be some differences, okay? And that's evolved over time as they adapted to their roles and things like that. Okay? But for the most part, the cellular machinery, okay, and the cellular processes are identical. There are a few differences, but there's more alike than there is dissimilar. Right? Now, what are some, if I was looking at like defining or naming the characteristics of a living organism, what are some things I would say all living things have or do this? What are some things I would say if I was making that list? If something is alive.
I have something else. Did you turn on the...
Yes, I did. Okay, so all living things produce waste, because they don't all poop, but yeah, they all produce waste for sure.
They need water.
They could all.
Or energy.
Okay, they would all need energy for sure, yes. Okay, either they're getting it somehow or producing it themselves, but yes, they would all need energy.
They all adapt to their environment.
Yeah, or they die, yeah. They would adapt or die, yeah. Okay, what else? Ability to reproduce. Okay, so we kind of got the big 3 there, okay? They all use energy, they all produce waste, they all reproduce. Okay, if I just look at those 3 qualifications, because we're missing one important one here. If I look at just those 3 qualifications, fire is a living organism. It can reproduce, it produces waste, and requires energy. Is fire a living organism? It can look like it sometimes, okay, but it's not. It's a chemical reaction. Fire is not alive because it's not made of what? Cells. All living things contain cells. Okay, that is the basis of the cell theory. If something is alive, it is made of cells, which is why there is argument about whether viruses are true living organisms. Okay? A virus has no cellular structures, carries out no metabolism, and cannot reproduce on its own. Okay? A virus actually doesn't meet any of the 3 characteristics of a living organism. Fire does a better job of meeting the characteristics of a living organism than a virus does. Okay? A virus works like this. A virus will attach to a cell, okay? Whatever type of cell it is designed to attach to, okay? It will inject through the cell membrane its own DNA, which will be absorbed by the cell. Now, what does DNA tell a cell how to do?
To do general stuff, mainly how to make more of itself.
Yeah, exactly. Okay? If I was gonna make a copy of you, okay, if I was gonna clone you, I need all of your DNA. And your DNA would tell my cloning machine, okay, how to make an exact copy of you, how to do all the processes you do, okay? All of that is encoded in your DNA. So when a virus infects a cell and injects its DNA inside, the cell absorbs it and it gets spliced in to the cell's own DNA. The cell can't tell that there's anything wrong because DNA is DNA, okay? And in that coding of DNA is a short little piece of code that says, make this. Okay? So the cell starts making this stuff. Okay? The cell thinks it's making something else, like let's say insulin or adrenaline. Okay? It's supposed to be making something it needs. So it makes a virus. And then looks at itself and goes, wait a minute. Weren't we supposed to make something? We're supposed to make insulin here. Well, do it again. Something got lost. Do it again. What does it make? Another virus. Cell does another check. Hey, didn't we order that? Order it again. Over and over and over and over again until the cell is full of viruses and bursts. Okay? It just creates a feedback loop and the cell just keeps making more and more viruses until the cell literally pops and the viruses escape. And then each one of those viruses goes out and infects another cell. Now, they probably don't all infect another cell, but they all have the potential at least to do that. Okay? Now, a virus, because it's not alive, okay, doesn't really survive all that long outside of a host cell. Okay? If it doesn't attach to a host cell within a short period of time, it simply breaks down. The body absorbs it, okay, or destroys it with looking at like your white blood cells and immune system and stuff like that. Okay? The reason a virus makes you so sick is because the way your body fights it is to kill the cells that are infected. Okay? That's why when you get a virus, you feel depleted, weak, okay? And then you have all the immune symptoms, or the immune system suppression symptoms, coughing, sneezing, runny nose, all that kind of stuff. Okay? That's why you're told, drink lots of fluids, okay? Eat soup, all that kind of stuff, because it helps to flush out the products of the body getting rid of its own cells. Okay? So in order to kill a virus, your body has to kill the infected cells. Okay? It can tell a cell is infected because it usually has a virus particle on the membrane, and that's what the body can look for. Okay? A cell that is infected is behaving differently. Okay? It has a different chemical signature, and the body can hunt it down and kill it. That's how immunity works. Okay? If you had chickenpox, for example, okay? Now I don't know how many of you actually had chickenpox. Most of you probably just got vaccinated for it. Okay? Back in my day, there wasn't a vaccination for that. Somebody in the neighborhood got chickenpox, everybody went over to their house to play. Then everybody got chickenpox, and then everybody was immune to chickenpox after that. Okay? It works the same, you just didn't have to get chickenpox, which is not pleasant. Okay? So once you have that, or you've gotten the vaccination for it, okay, your body can recognize that intruder. Okay? When it sees it, it already has cells that know how to kill it, it just produces them, and it goes out and kills it, and you barely get sick at all. Okay? That's how immunity works. All right? Problem with things like the flu and coronavirus and stuff like that is they mutate. Okay? So I could get the flu more than once in a flu season. So maybe I get it right at the beginning, and I get immune to that, but the flu virus mutates over the course of the flu season, and by the end of flu season, it's different enough that my body doesn't recognize it, and I get it again. Okay? That's why when you get your flu shot, you're actually getting a whole bunch of different strains of the flu. Okay? What the immunologists do is they just go, okay, we're seeing a lot of this kind, and a lot of that kind, and a lot of this kind. We're going to put those ones in the flu vaccine for this year because we think there's a high degree of probability the flu virus for this year will be like these ones. And that's how flu shot works. Okay? And most of the time they get it right. Okay? I was never big on the flu shot until a couple of years ago. I have not had the flu since getting All right. So if we're looking at, okay, the cell theory, the first point of the cell theory is all organisms are made up of cells. Okay. Some organisms are going to be unicellular, so single-celled, okay, and others are multicellular, like a cactus. Okay, so this would be like a paramecium. This lives in like scummy pond water. Okay, these are actinopods. They live in the sea, okay, in salt water, and then obviously cactus is our multicellular organism. Okay, so all living organisms are made of cells. Viruses, not necessarily by the truest definition a living organism. Second point of the cell theory. This is probably the most important one, so you might want to highlight it somehow. Okay? The role of the cell in your body is to carry out the basic functions of the organism. Okay? What we mean by that is this. Anything you do on a large scale, the cell does on a small scale. Okay? That would be respiration, digestion, metabolism, okay? Burning sugars, all of that kind of stuff, producing wastes. All of that's done on a cellular or small scale by your cells, and you're doing it on a larger scale as a full organism. So the cell carries out your basic functions. Okay? For an animal like ourselves, the basic functions are, okay, acquire food, digest that food, metabolize the chemicals in that food, get rid of the waste products that result from that, then reproduce. Okay? So those are the things the cell has to do, the basic functions. Right? And the last point of the cell theory, new cells are not created. There's no factory in your body producing more and more cells. Okay? Cells come from the division of existing cells. Okay? So even in a place like your bone marrow, where we often say cells are produced, okay, they're really just produced from cells that are in your bone marrow dividing. Okay, so your cells divide to produce new cells.
So if blood cells might be different in this way, I don't know, but but let's say you start bleeding, you lose some blood.
Yeah.
So do you permanently lose that amount of blood for the rest of your life, or how does it work?
You can replenish it because your liver and your bone marrow the cells within them can produce more cells. Okay? So there isn't just a factory where cells are assembled and put out. Okay? Your bone marrow is kind of a cell factory, but it's only because within your bone marrow are like stem cells which can produce new cells by dividing. Okay? So stem cells are undifferentiated cells in your body that simply divide and can produce cells on demand. But they do that by dividing themselves. Does that make sense?
Kind of.
Okay, so if I lose a lot of blood, okay, the first thing my body's going to do is try and replenish the volume. Okay, that's just the plasma. Okay, not necessarily the cellular components. Okay, so I need to consume a lot of fluids, get an IV, something like that, just to replenish my fluid volume. Okay, to get my cellular volume back, okay, my bone marrow and my liver will work hard over the next week, depending on how much blood I've lost, okay, to produce more cells. So if I go and, let's say, donate blood, okay, so I give a pint of blood, so like 454 mils, okay, of blood, okay, I go and give that away. I've given up about an eighth of my blood supply. Okay, I'm going to now need to have some juice, so I usually give you some orange juice, okay, get your blood sugar back up, get a little bit of volume back, okay, need to make sure I drink lots of water, and over the next few days, get some protein, okay, some red meat, things like that, so my body can start producing more blood cells. If I don't eat meat, then as a vegetarian, then I would have to eat more beans and legumes and stuff like that have protein, tofu, okay, stuff like that, in order to get those cellular components up so my body can make more cells. My cells can divide more rapidly and reproduce, make more.
So these stem cells have more DNA inside of them, which allows them to produce more, or?
No, they don't have more DNA. All of your cells have the same amount of DNA, except your gametes, which have half as much DNA as a normal cell. Okay, but your stem cells are just types of cells in your body that can produce cells on demand. Okay, by dividing. I don't know how else to explain that really. When you are an embryo, okay, so the day you were conceived, when sperm met egg, okay, all of the cells that are now you came from that cell. That cell divided over and over and over and over and over again to produce all of the cells in the body. But when it first formed, it was unspecialized. Okay? It was just a cell. Right? As it divided, it produced different kinds of cells as well. Okay, so during the process of development, okay, it's called embryology. Okay, the cell divided, and then certain parts of that, certain cells, when it gets to like 16 cells, you starting to see this cell is now going to become a liver. This cell is going to become the nervous system. This cell is going to become muscles. This cell is going to become the brain, right? And so you start seeing that differentiation. Embryonic stem cells are the best kind of cells for growing tissue from. Okay, I'm going to go over to stem cells before. Okay, there's different kinds of stem cells. Embryonic stem cells are the kind of stem cells that are actually banned in Canada and many countries because they involve, you have to fertilize an egg, you have to let it divide a bunch of times, get to a certain stage of its development, and then you harvest cells from it. Okay, so what do you do to that embryo? You kill it. Okay, that's as Catholics we believe that's not right. Okay, that is a living person. Once conception occurs, that is now a living person. It has all the potential to be a living person from a biological standpoint. That is an absolutely true argument. All of the DNA, all of the genetic material, all of the potential is there at that moment. Okay, that's why as Catholics we believe that life begins at the moment of conception. Okay, because it's all there. Right? So if you allow that thing to develop, that embryo to develop to a certain point, then you start harvesting cells, the embryo obviously dies. Okay? Those are, however, the best cells for producing tissue from. So in some countries where that research is allowed, what they're doing is they're using those cells to grow tissue that can then be transplanted onto a full-grown person. So if I was diabetic really badly, I could get pancreatic tissue grown from stem cells and then grafted onto my pancreas. And that would effectively remove my dependence on insulin shots because I would now have healthy cells that are producing insulin properly. Okay? You can grow skin for skin grafts, things like that. Liver tissue that can be grafted onto the liver of a person whose liver has failed. Okay, stuff like that. Now we're not growing like full organs. Okay, don't get the idea that we go, oh, we'll just go and make that liver, okay, and grow it and stick it in somebody. We can grow the tissue, so we can get a big, like a mat of cells that are liver cells that could be grafted on to a liver. Okay, it's all still in its infancy because a lot of countries are banning that research because of the implications of it. Okay, so cells come from the division of existing cells. I talked about that a lot longer than I intended to, but that's good. If you guys had questions, that's fine. Okay. All right, now that division process is pretty complex, okay? You think about everything that's in a cell, it needs to be copied, okay? And it needs to be copied flawlessly, okay? All of your DNA has to be copied before the cell can divide, because both new cells need all of that DNA in order to do everything, okay? So every time a cell divides, and they divide fairly often, okay? That process has to occur flawlessly. If it doesn't, the cell will die, okay? Or a cell that's produced will be missing some information or have duplicate information, okay? And either one of those situations is bad, okay? If this process doesn't occur properly very early on in embryonic development, or even in the formation of gametes, then the implications for the baby are dire, okay? For example, Down syndrome, okay? How many people know somebody who has Down syndrome? Okay. Down syndrome is caused by this process not occurring correctly very early on in their development, possibly even when the gamete, the sperm or egg, okay, was formed, right? For some reason, they get an extra piece or full copy of chromosome 21. Okay. Down syndrome is called trisomy 21. There's 3 copies of chromosome 21 when there's only supposed to be 2. Okay. The implications of that extra copy are all of the symptoms, all of the stuff that goes along with being Downs. Okay, there are lots of other trisomies. Many of them are even more severe. Like a person with Down syndrome can live a pretty healthy life, other than the physical and mental parts of that. But there's a lot of trisomies where that person will not maybe even come to term, which means they may not even be born. They may be a miscarriage. Okay, some of them are so severe that a woman might not even know she was pregnant. Okay, like they're that bad that the cells divide for a little while and then it just quits. Okay, And some of them, they can become full term and then they die shortly after born. They can be pretty severe to have a problem with the DNA. It can be very problematic. All right, so that process has to occur, and it has to occur pretty flawlessly. Okay, so development of the cell theory. Who's important? Okay, maybe not most important person, but the person who discovered cells in the first place was Hooke. Okay, now Hooke was actually a physicist, which is why physics is awesome. Okay? Why physics is the best science, and why all other sciences should bow down to physics. Okay? You've heard this before. I see people rolling eyes, but it's true. Okay? I mean, come on. Physics discovered the atom. No chemistry without that. Okay? And this guy who's just playing around because he's bored is doing an experiment with lenses. He's looking at the refraction of light and how the shape of lenses can change the path of light. In doing so, he develops something that can magnify things greatly and discovers the most important thing for biology and says, hey, look what I made for you. Now your science can be real. There you go. Okay, so Hooke develops the first microscope. Okay, now compared to what we use today, this is not great. It magnifies about 30 times. A good magnifying glass can do that. Okay, but at the time, this was a pretty big deal, right? And what he did with it was he was just trying to look at stuff. He put like hair underneath there, stuff like that, and then he sliced a wine cork and he stuck it underneath the microscope. Now, what's a cork? A real one made out of? Wood. Comes from a cork tree. So he puts it under the microscope, and this is what he sees. What is he looking at? Yeah, plant cells. Okay, now they're dead, okay, because the piece of cork would have obviously come from a tree that had long been harvested. Okay, but all he sees really are the cell walls. Okay, all the living parts of the cell would be in the middle, and because these cells are dead, it's not there anymore. Okay, but what he sees is basically the remnants of these cells, which is why he called them cells. They were empty little rooms. Okay, like cells in a monastery or in a prison. Okay, they were just empty little rooms. Okay, that's why he called them cells. But he discovered the basic unit of life by playing around with lenses. Okay, doing a physics experiment. Okay, he discovered those. So Hooke discovered cells. Okay, so he's a big deal. Now, after that, the development of the microscope takes some strange turns, and there's some weird shapes and styles and things like that, like this one that Anton van Leeuwenhoek made. Okay, it looks like a ping pong paddle, but actually what it does is you hold it up to the light, and the light goes through the lens. Okay, it goes through the specimen, then through the lens, and you can like put it on the screen a little further back. You get a nice light source, and that's how it would magnify things. Okay. And so looking through that, or just looking through the lens like this, okay, if you could change how big it was by moving it away from your eye, okay, you could see the organisms that were in it. Now, Leeuwenhoek's big contribution was he proved that cells weren't empty. He looked at living cells. Okay? He was the first person to view living cells. So he looked at things that were swimming around in the pond water. Okay? He looked at blood cells. He looked at sperm cells. I don't know how he got sperm cells from a bull. He was obviously very brave. Okay, so I would have to think that would be somewhat risky. Okay, but he looked at them, and his microscope could magnify up to 300 times. Okay, the microscopes we use can magnify up to 400 times. So, pretty good. He had pretty good equipment. All right, then 1839, these two guys come along, two Swedish scientists. Okay, Matthias Schleiden and Theodor Schwann. Okay, they come along and they are doing an experiment where their hypothesis is this. Okay, if cells are the basic unit of life and tissue from various organisms is viewed under the microscope, then the tissue samples will all contain cells. That was their hypothesis. Okay, they spent years looking at the tissue from different types of organisms, plants, animals, fungi, okay, bacteria, all kinds of things. And what did they find? Yes, without exception, every sample from a living organism contained cells. Those cells were all widely varied, quite different, but they all contained cells. Okay? So that was the cell theory. These guys developed the initial cell theory. Okay? Once they discovered that cells were in fact in all living organisms, point number one of the cell theory, okay, they started looking at what do cells do. Okay? And they found that cells carry out the basic functions of the organism. In a plant, they carry out photosynthesis. They burn sugar for energy. They produce proteins and fibers and stuff like that. Okay? They carry out the basic functions. And then they watched and saw that all cells have the ability, well, I shouldn't say all, but pretty much all cells have the ability to divide. And when they divide, that's the only way that they can reproduce. They never found any cases where cells just magically appeared out of nowhere. All right, so those two guys probably should know their names. I mean, there's really only 4 names to remember here. Hooke, Leeuwenhoek, Schleiden, and Schwann. Okay, those are the 4 you need to know. Now, as our technology improved, we were able to view cells closer and closer, magnify them even more. Okay, now we have electron microscopes where we can see individual tiny pieces of each cell, okay, in minute detail, right? And what we're discovering is that cells from different organisms have more in common than they have different, okay? And what that's led us to believe is that life on Earth likely originated from a common single cell ancestor. Okay? That the first living thing that appeared on Earth gave rise to everything else. Okay? Now, a little bit later on in the unit, I'll explain to you why that doesn't go against what you learn in religion class. Okay, I don't have time to go over it today, but I do devote an entire lesson to it later on in the unit. Okay? So I'm not being a heretic or anything like that. Okay? I'm telling you anything different than you would learn in religion class. Okay? All right, so all plant cells have similarities as well. They contain similar structures or types of cells that are all adapted to the same purpose. Right? So if we're looking at, okay, these 2 cells here, this is a bone cell, okay, or bone cells, and this is cells from the stem of a plant. Okay? The job of a bone and the job of the stem of a plant is to support the organism. Do they have a similar appearance or layout? Right? Generally circular, okay, generally kind of dense, okay, densely packed, things like that. Okay, all of that leads to being able to carry out these functions. All right, so earlier I said if I put a plant cell and animals together, okay, would they have more alike than they would have different? Okay, well the answer is a lot alike, right? They both have a nucleus. They both have rough endoplasmic reticulum. That's this blue stuff here. Okay, so this is the nucleus here. Okay, they both have mitochondria. Okay, these orange sausage-shaped things. Okay, they both have a Golgi apparatus. Right, they both have smooth endoplasmic reticulum, which is right here. Okay, they both have cytoplasm, which is the fluid that fills up the cell. Okay, they both have a cell membrane. They have a lot of things in common, and that's just the structures. Not to mention all the processes that each of those cells uses. Cellular respiration is identical between the 2 cells, is how they burn sugar. Okay? What are some differences? Well, plants have a cell wall, animals don't. Okay? That's because plants grow in one place and have to support themselves that way, whereas animals have to be able to move, and the cell wall would make them too rigid to move. Okay? Plants obviously have these green things called chloroplasts, which carry out photosynthesis. Animals don't have those. Okay? If we did and we felt hungry, we just have to walk out into the light and we wouldn't feel hungry anymore. Okay? We could produce sugar just by standing in the sun. Animal cells have little organelles called lysosomes that digest food. Okay? Break it down into amino acids and sugars and things like that, which can then be used for energy, whereas obviously the plant just makes the sugars directly. Okay? So there's some similarities, some differences, but for the most part, they have far more in common than what they have different. Okay, so that's why we believe this common single-celled ancestor idea. Okay, microscopes. All right, I'm just going to run across the hall and grab a microscope. I wasn't sure I was going to get this far, so I'm just going to go across and grab one here. Okay, so these are our light microscopes. Okay, you'll notice the way I'm carrying it. How many hands am I using? 2. Always use 2. All right. All kinds of stuff can happen in a lab, okay, and I've seen it happen. Somebody's just carrying it with one hand like this, and somebody bumps their arm, hits their elbow right on the funny bone. Okay, what happens when that happens? You let go. Okay, crash. $650. Okay, yeah, they're not cheap to replace. So carry them with both hands. Okay, one on the base, one on the arm. Okay, that's this big part back here, the 2 heaviest pieces of the microscope. That's where we want to bear the weight of the microscope. Okay, so those 2. Now, for the microscope, they all have dust covers. They're either Ziploc bags because the dust cover that came with them originally was torn, okay, or it's the original dust cover. Okay? Make sure that always gets put back on there. Right? Inside the microscope, sorry, on the outside of the microscope, you will see the power cord. Most of them have been spliced already, like this one. Okay? It's got lots of electrician's tape on it. Okay? Just be mindful of that. They're not new. Okay? So that's where you will get the power to supply the light, okay, on your microscope. Okay? You've got an on-off switch on the back. Okay, that will activate it. Okay, there's a rheostat on the side that can adjust the brightness of the lamp. Okay, and then underneath the stage, that's the flat part where the slide goes, okay, is the condenser diaphragm. That can also be used to change the amount of light that comes through. Okay, and by moving this little tab here, this is the condenser diaphragm. Okay, it's basically like an iris. Okay, I can actually not only change the amount of light coming in, but I can also increase or decrease the contrast. That is the difference between light and dark on whatever I'm looking at. It can really make something jump out. Okay, if I'm looking at something that's kind of hard to see, maybe it's not stained, so it's almost transparent. Adjusting this can just make it all of a sudden pop. Okay, so it's a way to adjust the clarity and the contrast of your image. These ones, different probably than the ones you used in junior high, have a movable stage. Okay, they have little dials on them that can move the slide around on the stage, and that's a lot handier than what you probably had to do, which is put your thumb on the slide and move the slide around, which inevitably moves the stage and puts it out of focus. Right? So this allows you to just move the stage from side to side without losing the focus. Right? So that's what those two little knobs do there. Okay, then we got the stage clip. Right? So you pull it out to the side, put the microscope slide in, it closes on its own, spring-loaded, okay, and it'll hold the slide in place. Okay, you have the two focusing knobs, the fine focus knob. Okay, if you look, when I'm turning this, can you see the stage move at all? Okay, it doesn't move the stage very much. It's for very, very fine focus. The coarse focus knob, on the other hand, moves the stage a great deal. In fact, it doesn't even make a full turn before it hits the stops on either end. Okay, so that's if you're obviously like way out of focus and you need to go fast. Okay, you can use that one. This one should only be used on the first two lenses. Once you get to the high power lens, that's the big fat blue one, big long fat blue one, okay? You don't want to use the coarse focus knob anymore because you can actually drive the slide through the lens. Okay, see how the lens is now down below the level of the stage clips? Okay, so you can actually break the slide and scratch the lens by using this on the high power lens. So you should only use the fine focus on the high power lens. Okay. All right, which lens should you always start on? Yeah, the first one, which we call the scanning power lens. We call it scanning because you use it to scan the entire slide and look for the specimens that you need to find. Okay, then you have the low power lens. That's the middle one. Okay, it's got a yellow band on it, and then the high power lens is a blue line on it. Okay, the eyepiece at the top can be moved. Okay, it's great if you're working with a partner. One to be on one side of the table, one on the other side, allow them to pick up the microscope and turn it, because every time you pick it up and set it down, it goes out of focus. Okay, this way all you have to do is turn the eyepiece around. Okay, obviously one of our COVID procedures will be that before the next person looks through it, it gets wiped down. Okay, I know that seems silly, but one of the ways to contract it is apparently through the conjunctiva of the eye. So we don't want to do that. Okay. I'm never going to ask you to label a diagram with a microscope, by the way. Okay? Just know what the parts are, so when I say, oh, do this, you know what I'm talking about. Okay? You don't look at me like I'm speaking Greek. All right, so microscopes are always carried with one hand on the arm, one hand supporting the base. Microscopes are always returned with the lowest power or scanning power lens in place. Okay, so always make sure that one is in place when you return it. Okay, when using a microscope, always start with the lowest power and move up, and only use lens paper to clean the lenses. That's really important. Okay, if your lens seems dirty, or you can obviously see a fingerprint or an eyelash or something like that on it, come up to the front and get a piece of lens paper. It's special paper that gets used one time to clean off the lens without scratching it. Okay, because I've seen people do this. They're like, oh. And they like wipe it with their thumb, or they take a shirt and they're wiping. There's all kinds of grit on your skin and your clothing. And depending on what fabric you're wearing, even that can be harmful to the lenses. You put a scratch on a lens that magnifies 400 times, what does it look like? a tiny little scratch becomes a gaping chasm. Okay, and you can't see anything through that lens anymore because you've magnified that tiny little scratch. Okay, so we want to make sure we're always using fresh lens paper anytime we need to clean the lens. Okay, so other stuff to do with the microscope. Okay, we already talked about all that stuff. Okay, this you'll need to copy somewhere, sheet of paper, something like that. This is how we estimate the size of an object. I don't know if we're going to get through this, but we'll try. Okay, so this diagram is showing you the field of view for your 3 lenses on the microscope. Okay, the field of view is the area that you can see when you look through that particular lens. So, our scanning power lens, the little red one, okay, magnifies 40 times. All right, so anything I look at through there is 40 times larger, or appears 40 times larger. The field of view is 4,000 micrometers. Okay, that's actually 4 millimeters. Okay, but it's 4,000, and then it's mu. That's this character here. It's the Greek letter M. Okay, mu meters. Okay, you can just use a U for that. Okay, that's generally what we do. Okay, just use a U for mu. Okay? We don't obviously use M for micro because we already use it for milli, and we can't use it twice. That's why we use the Greek letter M instead. Okay? So 4,000 micrometers is the distance across my field of view. Why is it important to know that? It helps me to estimate the size of the objects I'm viewing through the microscope. So if I know that the distance from one side of the circle to the other is 4,000 micrometers, approximately how big is that? 1,500 to 2,000 micrometers. Takes up about half. Okay, it's just an estimate. I don't have to be exact. Okay, but that's about 1,500 to 2,000 micrometers in size. Okay, that's why it's important to know your field of view. Your low power lens, that's the middle one that's got the yellow band on it, okay, magnifies 100 times, and its field of view is 1,600 micrometers. Okay, which means now if I take this object that I was looking at on scanning power and put it here, it suddenly takes up a heck of a lot more of my field of view and probably looks a lot bigger. Okay? Which means I also did a pretty darn good of estimating its size between 1,500 and 2,000 micrometers. Okay? When I go up to the high power, my field of view goes down again. Okay? My magnification goes up, but my field of view goes down. When I move up to high power, I'm magnifying 400 times. So anything I look at is 400 times larger than it actually is. But I only see a 400 micrometer area. So now I take this thing over there, and now I'm looking at individual parts of it. I can't even see the whole thing anymore. Okay? I'm just looking at a small piece of it. Okay? So why do I want to start on the scanning power lens if I see all the detail on this lens? Okay, you're on the right track. Let's say I go to a football game and I know my friend is on the other side of the stadium. Okay, and he's wearing a bright yellow shirt. Do I want to look for him using binoculars or a telescope, or do I want to first scan the other side for something yellow with my eyes? Okay. If I just have a telescope, I'm looking at each individual person on that side, thousands of people one at a time, in order to find my buddy. Okay? That's not realistic. I want to look for something yellow and then go, all right, now I know where to look, okay, and focus in with something bigger. Sort of make sense? That's what we're doing. We always start scanning that way. Okay. We'll call it there, guys, and we'll talk more about handling of the microscope and do some pre-lab stuff probably tomorrow. Okay.